Oh. Hi, my name is Lila McLendon.
My name is Karen Ajay.
My name is Jenny Munoz.
And our presentation is on whether fabrics such as cotton, tulle, and silk can protect yeast cells from UV radiation. Our question was, can fabrics be forms of protection for yeast cells against UV radiation?
So for background information, UV radiation is known to harm the DNA of skin cells, and too much exposure could lead to skin cancer. It has been discovered that sunlight is the main source of UV radiation. However, UV light can also come from artificial sources. The CDC has shared that tanning beds, some halogen, fluorescent, and incandescent lights, and lasers are alternative sources of UV radiation. The amount of exposure to UV light depends on the strength of the light from the source, the time of exposure, and if the skin is protected. In this experiment, we will expose yeast cells to various amounts of UV light with several fabrics as protection. We will also measure the amount of UV damage after the protective use of different fabrics such as silk, cotton, and tulle. The yeast cells in this experiment are used to simulate human skin cells. The hypothesis. What is your hypothesis? With silk as protection, more colonies of yeast cells will be able to survive exposure to UV light. However, with the tulle used as a least colonies will be able to survive the exposure to UV light. What do you predict will happen? In this experiment, silk was observed to preserve the most yeast colonies compared to the other fabrics due to its strong stitching, while the tulle was observed to be the least protective fabric on account of its net-like material. Is this a multiplex experiment? This is not a multiplex experiment because there is only one independent variable, and that is the type of fabrics, silk, cotton, tulle used to shield the yeast colonies from the UV light. Variables. What is the independent variable? The various types of fabric, tulle, silk, and cotton. What are the dependent variables? The number of yeast colonies that survived and the survival percentage. So basically the measure of the yeast colonies that survived. What is being held constant? The type of agar on the plates, UV source, time of UV exposure, and time in incubation. Lila, I thought that was you. Or is that me?
That's still you, yeah.
Okay. So the procedure that we went through for this experiment first off is the dilution process. So to begin diluting, first each tube is filled with 990 microliters of water, and 10 microliters needs to be diluted into each tube. Undiluted yeast cells are vortexed and micropipetted into the first tube to get the first dilution. Next, 10 microliters is transferred into the second tube to create a 10 to the minus fourth dilution. Lastly, 10 microliters from the second tube is transferred into the third tube to create a 10 to the minus fifth dilution. After dilution, 9 YPD plates are exposed for 30 seconds to the UV light. The agarose half bottom of the plate is labeled with the time of exposure. The culture is mixed well using the vortex, and then the yeast cells are then plated on the YPD plates. 100 microliters of dilution are plated on 9 different YPD plates, and then the next step was to expose the yeast to UV radiation with the protectant, the fabrics, the different types of fabrics between the light and the yeast. The fabric, tulle, silk, or cotton is placed over the light box, a flat surface with the UV light beneath it. And then we laid the plate on the agar side onto the fabric. Each fabric is exposed with the yeast for 20 seconds each, and then close the UV light box lid when we are using the UV light source. Finally, the plated yeast are placed into a brown bag and placed into a 30-degree incubator for 2 days And then the next week, we check for the number of colonies on each of the plates and further use the data for calculations.
So our results. So these are the results that we got from our experiment. So these are the results from experiment 1, in which we exposed the yeast cells to the UV radiation without fabric. So here you can see that there were 146 colonies, then it decreased at 10 seconds to 74, it decreased at 20 to 51, and then at 30 seconds, it decreased to 16. The ones over here, measurements over here, are from the measurements over here, sorry, are the amount of colonies that were present before we exposed the yeast colonies to UV radiation using the fabrics. So at the beginning, there were 149 colonies of cotton. There were 148 colonies, or yeast cells, sorry, of tulle, and then silk, there were 143 yeast cells. And then as we went during the experiment, as we continued to expose them for more UV light exposure, they continue to decrease. Here are calculations of survival rate. So based on the numbers that we had on the previous table, we then calculated survival rate. So for without fabric, at the beginning, there was 100% survival rate, then it decreased to 51%, then 35, and then 11. So at 30 seconds, there was only 11% survival of the yeast colonies. For cotton, similarly with the without fabric, we had a 100% survival rate at the beginning. It decreased to 98, and then 97, and then 77. It's the continuation of the calculations. So for tulle, at the beginning, again, there was 100% survival rate. It decreased to 57%, then 41, and then 27. And then lastly, for silk, at the beginning, there was a 100% survival rate, and then it decreased to 84, 66, and then 50%. So here is our graph with the data of the percent survival rate, and then in comparison to the time or seconds of UV exposure. And then we also calculated our LD50s. So without fabric, when the 50% of the population was still surviving, it was at 10 seconds. For cotton, the LD50 was about 40 seconds. It's not currently shown here because our experiment, we didn't go past 30 seconds, but we estimated it to be about 40 seconds for when the population is 50% surviving. For tulle, the LD50 was 15. And for silk, the LD50 was 30 seconds. So for the results, the yeast cells used for the protection of each fabric having the same time in seconds of UV exposure, the agar of each plate was constant. The control group was the yeast cells exposed to UV light without the use of a fabric protectant. The experimental groups of which a fabric protectant was used, such as cotton, tulle, and silk, had a higher survival rate percentage throughout the seconds of UV exposure to light, which were 0, 10, 20, and 30 seconds, compared to the controlled group. In other words, the experimental groups of yeast cells seem to gradually die off more during the UV light exposure rather than die off quickly like in the control group without the fabric. As the graph illustrates, which we previously showed, the tulle was the least capable protectant of the three fabrics. When comparing the LD50 of each fabric, tulle has the lowest time in seconds at the 50% survival rate, meaning that the yeast cells were dying at a faster rate during the UV exposure. However, at the LD50 of cotton, the time of UV exposure was the highest, revealing that the dying rate of the yeast cells was the lowest, making cotton the most protective fabric, and silk the second most protective fabric in the experiment. Using the tulle, the population of the yeast cells reached a 27% survival rate at 30 seconds, while the populations of yeast cells using the cotton and silk still had at least a 50% survival rate.
So the conclusion, Does the results support or refute your hypothesis? Okay, the results in the experiment partially refute the hypothesis, sorry. Since we anticipated silk having the highest survival rate. However, according to the results of our experiment, cotton had the highest survival rate percentage. At the 30-second mark, Toll had a 27% survival of yeast cells remaining, and silk having 50% yeast survival rate. Lastly, cotton had a 77% yeast survival rate. Furthermore, our hypothesis was also partially supported through the results of the toll as a protectant, because we hypothesized that toll would be the less protective fabric. These survival percentages compared to the 30-second mark of the yeast cells without fabric protection was much higher, showing that fabrics can act as protectant against UV light exposure. Conclusion continued. These are some implications or bigger meanings of the results. The bigger meaning of the results are that certain fabrics can be taken into consideration when exposed to UV light sources. Based on the results, Toll allowed enough UV light source to kill all of the yeast cells, and this primarily because of the net-like features Toll has, allowing the holes to give UV light access to the yeast. However, cotton was the most capable of protecting more yeast cells compared to the other fabrics because of the quality stitch and cotton nature. These characteristics in cotton made it difficult for the UV light source to kill more yeast cells. Therefore, fabrics worn where there is UV exposure should be taken into consideration because some fabrics are more protecting than others. Conclusion continued. What additional experiments could be done to follow up these results or expand this study? To expand this study, other fabrics could be tested with the UV light and yeast, such as polyester. To expand the study, if polyester is used, this experiment could determine if it is more or less protective than cotton. The control, independent, and dependent variables should all be the same. The dilution process would be conducted again to create a 10 to the -5th dilution and spread on plates and incubated. After some time, the plated colonies of yeast would be exposed to the UV light source using polyester as a protectant and the data collected. The results would be compared to these results and so forth. And these are our references.
Well, I hope you enjoyed our presentation on using fabrics as a protectant to protect UV cells during exposure to UV light. Thank you and have a great day.
Have a great day.
Bye.
Bye.